as mentioned, my name is Vishnu, and my research is about the synthesis and characterization of novel levoglucosenone polymers. And so let's go ahead and get started. So my research was conducted at URD ABI, which is the Industrial Agrobiotechnologies R&D unit of AgroPeritech, which is a major research university in France. Specifically, the institution that I did my research at is based in Lens, which is the largest city of the Champagne region in France. And that particular research institute is part of a complex that involves Europe's largest biorefinery, as well as the variety of companies that you can see in the picture on the bottom right that are associated with that biorefinery. So, first, I'd like to name everyone who helped me there. So, my supervisor and mentor, Sammy, my graduate student, Enzo, who I did my research directly under, the lab director, Florent, and of course, the University of Florida, who I did this research program through. So specifically, my research was conducted through an REU, which was done through a partnership exchange between the University of Florida and a variety of French universities, including the one that I researched at. So some of the experiences that I did during my research internship, in addition to the research itself, were visits to some of the other facilities in the area. This includes Givaudan Active Beauty, which is a company that manufactures active ingredients for cosmetics using biotech. So Givaudan is one of the world's major fragrance manufacturers, but this subsidiary manufactures cosmetic ingredients like hyaluronic acid, which I imagine many of us may be familiar with, and also to Cristal Union, which manufactures ethanol and carbon dioxide gas for beverage production from wheat. So, in fact, the lab that I was researching at was surrounded on all sides by massive wheat fields, and that's because this wheat was being used to produce these chemicals. In the picture you can see in the center, that's me with the rest of the students in the University of Florida IREU program. So there were two other students with me at the site in Lens. You can see you can see those two in the leftmost picture, and the remaining students are all visible in the picture in the center. So we were split among four different French cities, researching at four different universities. So, since my project is about polymers, I want to give a bit of a background about polymers since I know that this topic isn't something that most people are familiar with. There are a bunch of different types of polymers, and they can be represented by the blue picture that you can see in the top right corner. Essentially, these are sorted by how strongly the different chains in the polymers are stuck to each other. So if you think of it like the jumble of wires that your earbuds cord can tangle into, linear polymers would be wires just sitting next to each other, easily separable, and a network polymer would be when you have 5 or 6 knots and it feels like you can never separate the wire. So depending on how well the polymer is linked, it has a variety of different properties, and I was trying to optimize for certain properties. One of these properties is called the glass transition, which is a property that certain types of materials called amorphous materials exhibit. And essentially, this is a difference in texture and material hardness that happens when heating, but it's distinct from melting or freezing that you would see with things like water or crystalline materials. I did my research using some techniques called DSC and TGA, which involve heating substances in different ways and measuring what happens to them. Essentially, DSC allows me to measure this glass transition that I mentioned, and TGA allows me to determine overall how thermally stable whatever material I'm testing is. So now, what is levoglucosenone? I imagine that's a question that many of you may have, since that is one of the big scary words in the title of my research project. Levoglucosenone is this very interesting molecule we can see here on the right, and I'm going to be calling it LGO from now on. Levoglucosenone, or LGO, is produced from cellulose. Cellulose is what most plants are made of, and cellulose is produced in large quantities as an agricultural waste product because humans cannot digest cellulose, but it's part of a bunch of plants. So using something called the Furacell process, cellulose is commercially converted to LGO on a large scale, which makes LGO available easily as a bio-based feedstock for a variety of chemistry. Because of all of these different sites on LGO, which are identified in the different colors in the top right picture, a variety of chemical reactions can be performed on it. And that means that the molecule has a lot of potential as a feedstock for making things like plastics, drugs, anything really, which are normally made with petroleum-based feedstocks, which we know are not renewable and also typically bad for the environment in how they're extracted. This is why we call LGO a platform molecule, and because of this interesting properties that it possesses, many research groups have done research on it, including those at ABI, the research institute I was part of. So the figure 3 in the top right shows some of the various monomers that have been derived from LGO. And just for context, a monomer is the small molecule that is combined repeatedly to form a polymer. So essentially, it's like a single paper clip, and you could link a bunch of them to form a chain of paper clips. So this picture shows some LGO-based monomers, but you can clearly see that they are not the same as plain LGO, which is a much simpler molecule. So my research involved the development of novel materials using LGO as the co-monomer instead of converting LGO to a different molecule first and using that new molecule as the monomer. And the advantage of doing this is that it simplifies the whole process, which means reduced costs and a lower environmental impact. Essentially, we would have to do less chemistry, use less energy. And I tested this using a variety of techniques, including TGA and DSC that I mentioned before, as well as NMR and FTIR spectroscopy, which are chemistry techniques that allow us to essentially see molecules, which of course aren't visible with the naked eye. And this other technique called gel permeation chromatography, which involves passing these polymer mixtures through a bed of little beads, which would separate the polymers based on size. Essentially, the bigger pieces of polymers can't travel through the beads as fast, so kind of like a sieve. What I ended up finding is that I was able to develop novel materials using LGO as the co-monomer, and I was able to observe a facile polymerization, basically nothing too complicated, mild conditions, no excessive use of heat, pressure, or toxic or dangerous reagents. And these materials exhibited a wide range of glass transition temperatures and thermal stabilities, which essentially means that they hold a lot of promise as a new family of materials. Overall, I conclude that LGO can serve as a cost-effective, sustainable monomer platform, which is great news for those of us who are trying to create renewable polymers that are free of oil-based materials. So here's a quick video showing GPC, and I made a couple of videos like this to post on the Science Influencers Instagram, as well as other posts that just involved pictures of what I was doing and where I went to. So here you can see me preparing a sample, doing a bunch of little manipulations, sped up really fast. I'm shaking some stuff and feeling stuff, filtering stuff. And this is what my days in the lab usually looked like. I was doing a variety of experiments like this, testing things, going to a bunch of different machines in different rooms, and logging my data to analyze it. So what I learned from this internship was quite varied in scope. I came into it knowing some technical skills, but most of my background wasn't in chemistry as I am a biomedical engineering major, and most of my past research has been more biology related. So I learned a variety of new things that could be applied to both chemistry and material science, and those are listed here. And this internship also led to me developing a variety of professional skills, such as improved lab competency, project management, professional communication, and of course, the kicker is science communication, which would be helpful to me in my professional career in a variety of ways, as I've listed there. And as part of the RU program, I will be attending the American Chemical Society conference in Atlanta in March, and I'll be excited to present my research there along with all of the other students who participated in this program with me. So that concludes my presentation. I'd like to thank everyone for listening, and please let me know if you have any questions. Thank you so much. And of course, I would like to thank everyone who assisted me in this. So that includes Drs. Leggett, Gastel, and Wingenbach, as well as Dr. Tammy and also Valeria, who were the program coordinators at the University of Florida, as well as Florent, Sammy, and Enzo, and everyone else at URD ABI. Thank you so much.